So our Frank Brooks state-of-the-art lecture today will be Dr. Timothy Wang. And Dr. Wang is the Silberberg Professor of Medicine and Chief of the Digestive and Liver Diseases at Columbia University Medical Center and the Director of the Tumor Microenvironment GI Cancer Program at Columbia. He is currently Chair of the American Gastroenterology Association and received his BA degree from Williams College and his MD from Columbia College of Physicians and Surgeons. He has a faculty appointment at MGH, where he was there for 10 years, and the Associate Chief of GI at MGH prior to moving to the University of Massachusetts as Division Chief in 2000, and then became the Division Chief at Columbia in 2004. He has over 260 peer-reviewed publications. He's been NIH funded for over 25 years, NCI funded for over 10 years. We are extremely honored that he has taken the time to come to this American Pancreatic Association and share with us his research in pancreas cancer and also his insights in the tumor microenvironment. So let us welcome Dr. Timothy Wang from Columbia University.
Thank you very much, Owen. It's a really a great pleasure to be here in Miami at this meeting, and it's been a great meeting, and I've really learned a lot. And I look forward to speaking about several of the projects going in my laboratory. So just as a brief overview, I'll start off by discussing the role of inflammation in pancreatic cancer. Inflammation in cancer is something I've worked on for a number of years. Then I'll talk a little bit about progenitor cells within the pancreas, and in both areas I'll include some unpublished data that we hope to publish soon. And I'll end with our discussion of the role of nerves in pancreatic cancer, something we've been working on now for about 4 or 5 years. But I thought I'd start off by paying a little bit of homage to Frank Brooks, for whom this lectureship is named. He was an MD, a gastroenterologist, Chief of GI at the University of Pennsylvania, a great institution, great program. And so I guess he and I share a few things in common, including we've both been president of the AGA. He is much more renowned. He won the Julius Friedenwald Medal as well. But he also was a renowned GI physiologist and was interested in both gastric and pancreatic exocrine function. So I started off working primarily on gastric biology, gastric disease, but I've switched over a lot of my laboratory now to pancreatic cancer. So inflammation and cancer, we've heard a lot about this at the meeting, this association, which goes back to the 19th century. Rudolf Virchow first described the presence of white blood cells, leukocytes, in cancer. And I still remember from my years in training when Harold Dvorak described cancers as wounds that do not heal, sort of a chronic inflammatory regenerative lesions. And as a gastroenterologist, it turns out that many of the cancers that we see are strongly linked to inflammation. And I think the point here is not only that inflammation is something that promotes the cancer growth, but also it's something that's present very consistently in the pre-neoplastic lesions. So in Barrett's esophagus and H. pylori, atrophic gastritis and metaplasia, chronic hepatitis, of course. Many of these are infectious diseases. I started off working on gastric cancer and Helicobacter pylori. And for me, that sort of became a paradigm for this sort of gastritis to metaplasia, dysplasia sequence as described by Pleo Correa. And pancreatic cancer has been sort of a bit of the outlier, the unknown entity in terms of how important is inflammation in not only the progression of pancreatic cancer, but its origins as well. And that's something that we sort of became interested in and have worked on in recent years. So we know that chronic pancreatitis is one of the strongest risk factors for pancreatic ductal adenocarcinoma. But when we think about this, we think about it as sort of the obvious clinical chronic pancreatitis with recurrent bouts, fibrosis of the pancreas, chronic calcifications. Those are the sort of patients we think, clearly they have pancreatitis and they are at risk for pancreatic cancer. But is it possible that there may be subclinical pancreatitis in many, if not most, patients in the preceding period? And how do KRAS mutations arise anyway in an organ where proliferation is quite low, as I'll describe? So we do know that this inflammation can lead to changes in the epithelia, changes in the stroma, induction, as we've heard, of acinar ductal metaplasia. As a biologist, the other thing we sort of have observed is that these mouse models with KRAS mutations, although they do rarely go on to pancreatic cancer without any inflammation, it's much faster in our laboratory when you use just a touch of cerulean. I think it was Ashok who first told me about this cerulean model of pancreatitis, and I go, what, frog CCK? But it turns out it works very, very well. And so we've been thinking now as chronic inflammation is clearly a promoter of cancer, but possibly involved in the initiation of cancer, and perhaps is it something that's really an essential cofactor for the development of pancreatic cancer. So I'll talk a little bit about interleukin-1 beta, which is a pro-inflammatory cytokine that's associated in terms of the signaling pathway with toll-like receptors. There's the TIR family of receptors leading to activation of MYD88 and other pathways, and it's clearly an activator of NF-kappa B pathways. It's also a downstream target of NF-kappa B, so it's clearly linked in very tightly to the regulation of numerous cytokines and chemokines, including interleukin-6 and interleukin-8 and so on. So, we have studied this initially in stomach cancer and also in Barrett's esophagus, and so what we found is that you overexpress IL-1 beta, it strongly induces cancer in the absence of any other intervention. So we looked into pancreatic cancer and the role of IL-1 beta, and I guess as shown in this slide, the one thing I'd say, it's upregulated in both human and murine models of pancreatic cancer, shown here in KPC mice and KC mice. It's much higher than in wild-type mice. It's associated with much worse prognosis. So patients with high levels of IL-1 beta, do not survive, as well as those with lower levels. So we went on to develop this, and this is over 10 years ago by Frédéric Marache, a French postdoc, this elastase IL-1 beta mouse model. And this turned out to be a very nice model of chronic pancreatitis. I think it was the first transgenic model of chronic pancreatitis. But unfortunately, even though they developed loss of acinar tissue and some ductular metaplasia, they did not progress at all to pancreatic cancer, and even showed no tendency even with the addition of carcinogens. So it shows you, to some extent, comparing it to stomach and esophagus, that the pancreas is a little bit different, and the factors that are involved in the initiation of pancreatic cancer in experimental models are still challenging. But clearly, we know that pancreatic cancer is associated with KRAS mutations. So we asked the question more recently, what happens in this elastase IL-1 beta mouse if you cross it to a PDX1 lock-stop-locks KRAS mouse model? And in fact, what you saw was a remarkable and very fast progression to pancreatic cancer, most often within a year. And so almost all the mice eventually over a year and a half or so develop pancreatic cancer. This is a survival curve that shows KPC mice here, wild-type IL-1 beta, and KC mice here. And so the addition of IL-1 beta and KRAS leads to accelerated mortality, and at 1 year, a much higher rate of pancreatic ductal adenocarcinoma. And in fact, many of these mice actually developed metastatic disease with mets to the liver, as shown here. So the combination, which proved to be quite synergistic, and again just highlights this idea that KRAS alone, in the absence of this set of inflammatory insult, doesn't do that much. So one of the questions we asked then was, how does this chronic inflammation driven by overexpression of interleukin-1 lead to the development of cancer? And so the first question was, what is the effect on the epithelial compartment, and particularly the acinar ductal compartment? And so if you look at the rate of proliferation in the IL-1 beta mice, it went way, way up from low levels in wild-type and IL-1 beta mice, moderate levels in KC mice, but the combination led to very, very high levels of proliferation, and as shown here with Ki-67 staining. The other thing we found was there was a much increased rate of spheroid formation. So this is the classic spheroid formation in major gel non-adherent conditions. And so again, you can see that this is in the KC mouse, not wild type it should be, with IL-1 beta, you saw a much higher rate of spheroid formation, which could be blocked by a receptor antagonist, IL-1 receptor antagonist. So probably IL-1 beta itself was doing something in terms of driving stemness or proliferation in vitro. We began to look at the effects of IL-1 beta in the microenvironment, and as you'll see here, we looked at a number of different types of immune cells. Many of them have been described in previous talks in this meeting. And so what was particularly interesting about this particular model was, although a number of immune cells were increased in the setting of IL-1 beta cross to KC mice, the biggest increase by far, as you can see here, was an increase in B cells, which was surprising and striking. And this is staining with B220, showing a comparison of KC mice to KC mice crossed to interleukin-1 beta mice. And the B cells were increased in the circulation and as well as in the pancreas tissue itself. So at a marked increase in B cells, not really a function that's been ascribed previously to interleukin-1 beta, and we're still investigating all the downstream pathways that link IL-1 beta increase to the increase, for example, of chemokines and cytokines that may attract B cells. But the B cell finding was sort of interesting. Most of the focus on tumor immunology has been on what's keeping CD8 T cells out of the pancreas, what are the suppressor cells, primarily MDSCs, Tregs, macrophages, suppressive macrophages. But B cells have also been described, and I think going back to now 10 years ago, Michael Caron described them in prostate cancer, and more recently, Lisa Cousins has had quite a series of papers describing these sort of suppressor B cells, which are really called B-regs. So we examined whether or not these B cells were in fact functional in terms of stimulating the growth of pancreatic cancer. So there's two ways it could do it, as we talked about. One is suppressing the T cell response, and the other is actually direct effects of many of these cell types, myeloid cells particularly, but perhaps also B cells, on stimulating directly the growth of pancreatic epithelial cells. And so this, on the left, we showed the slides of adding B cells to our sphere culture from KC mice, and demonstrating, in fact, that these were directly stimulating the growth of these pancreatic spheroids, KRAS mutant pancreatic spheroids. But the other was the effect on CD8 T cells, and this is a LysoSpot assay co-culturing CD19 positive B cells with the CD8 T cells. And what we found, of course, is that if they came from the KCl1 beta mice, they were in fact much more suppressive of both T cell proliferation as well as gamma interferon secretion, as shown here. So to show that these B cells are in fact tumorigenic, we then carried out adoptive transfer studies, taking B cells from these KCl1 beta mice bearing tumors, and then giving them to sort of, and as a control, B cells from wild-type mice, and giving them then to KC mice. And as you can see here, with this sort of regimen of injecting a series of 4 injections over time, and then looking at the development of pannins, as shown here histologically, as well as scoring in terms of pannin area, the transfer of B cells markedly increased the development of pannins and some pancreatic cancer in these animals. And finally, as a therapeutic target, would targeting these B cells actually be a therapeutic approach in terms of suppressing tumor progression in these KCl1 beta mice? And so we used a weekly injection of a combination of antibodies, anti-CD19, B220, CD22, and CD20, and demonstrating the complete sort of depletion of the B cells here. And in fact, the depleting B cells did improve the progression of pannins in these mice with markedly reduced pannin formation in the KC-IL-1 beta mice. So we're still sort of investigating exactly all the links between IL-1 beta and B cells, but I would say everyone's been looking for potential targets to improve, for example, immune therapy, and what are the immunosuppressive cells. And what we're learning, of course, is that there's multiple immunosuppressive cells, and it's going to take probably more than one target, and that may vary from patient to patient, including which cells really are blocking tumor immunity in pancreatic cancer. So now I'm going to switch now to the topic of progenitor cells in pancreatic cancer. And, going back years, we've been thinking for years as pancreatic ducts is the primary origin of pancreatic ductal adenocarcinoma, mainly because, in fact, these cancers resemble, aberrant ducts. But in recent years, we've been thinking more about other cells of origin for pancreatic cancer, including potentially centroacinar cells or acinar cells themselves. And we've heard a bit about ADM, the idea that these cells can transition into a duct-like phenotype, and that's very, very clear. But I guess the question regarding acinar cells really is there some sort of progenitor or stem-like cell within the population? Many have thought for a long time of acinar cells as sort of a homogeneous population, but as we start to do more and more single-cell RNA-seq, we know that most cell populations are not homogeneous. But the question about a true stem cell, I think so far most are convinced there's not a true stem cell. This would suggest maybe they're only progenitor cells. But there's also this concept of a facultative progenitor stem cell, which perhaps only becomes active or shows lineage tracing after injury, a sort of a reserve quiescent cell, which we've sort of been interested in as a potential cell of origin for pancreatic cancer. And so now I'll tell you a little bit about double cortin-like kinase 1, DCLK1, and we've heard an abstract presentation earlier from Courtney Houghton's group about this particular enzyme, which is a C-terminal serine kinase. Still being understood, was originally described as a marker of radial glial cells, precursors of neural stem cells in the central nervous system. But in the last 10 years, it's become of interest because it was shown to be expressed in the gastrointestinal tract, in the intestine, the stomach, where we began studying it. And it actually was Courtney Houghton who convinced me to start working on this nearly a decade ago. And so we were sort of interested in these cells. I think we now, in the GI tract, it's pretty clear that most of the DCLK1 positive cells are tough cells, which are not stem cells, perhaps as originally thought. But in the pancreas, it's a little bit different, and I'll describe this. I mean, there are, in fact, DCLK positive tough cells within the pancreas, very rare, which expand during injury and inflammation and pan inflammation. But there are also these DCLK like acinar, centroacinar cells, which we identified using this construct. This is a DCLK1 back Cree ERT construct that we generated that tagged a very rare rare population within the pancreatic acinar compartment, probably as shown here, 0.5 to 1% of the total pancreatic epithelium, so a very rare cell, which was long-lived as we followed it over time after tamoxifen induction, perhaps 2 years without much of a decline in the number of cells. But also completely quiescent, did not really expand, no BRDU uptake, so clearly not an active classic stem cell. But we were still interested in these cells. In fact, when we isolated them, single cells, what we found, as shown here in terms of spheroid formation, is they were much more active in spheroid formation compared to other acinar cells within the pancreas. And you could actually see lineage tracing in typical sort of spheroid assays using a non-isolated pancreatic epithelial spheroids. In addition, if you knocked out DCLK cells using a diphtheria toxin approach or DCLK1 kinase inhibitor, what you found was that they were much less active in able to form spheroids. So this was a set of a cell that had some stem progenitor-like features, even though it was relatively quiescent and seemed to be inactive during normal pancreatic homeostasis. So then we asked the question is, what is the role of this particular cell in response to injury during pancreatic regeneration? So we used our Cree ERT mice and crossed it to a diphtheria toxin receptor or a DTA mouse, and then ablated the DCLK cells using tamoxifen induction, and then used a fairly severe cerulean regimen. And so what we found was that in response to a cerulean-induced pancreatitis, there was very limited ability of the pancreas to regenerate after the injury. As you can see here, the comparison of a control pancreas treated with several doses of cerulean over 2 days compared to the diphtheria toxin ablated DCLK cell ablated pancreas. And in fact, the mice had a pretty significant 40% mortality over the 7 to 10 days, and the pancreatic weight was much less. So although not an active stem cell, this cell seemed to be important for pancreatic regeneration. And we went on to look at, confirm that was true using a variety of injury models. And as shown here, I think I have 60% partial pancreatectomy, bile duct ligation, pancreatic duct ligation. We also looked at cerulean models, of course, and radiation models. And what you see is that even though these cells remain quiescent through the lifetime of the mouse, normally, in response to injury, there is marked lineage tracing. Whole sections of the pancreas were generated from these single cells. So this is a quiescent cell that is very, very important in the response to regeneration. And of course, then our thought was that this can function as not as a cancer stem cell in that sense, but a cancer-initiating cell. Steve Leach and Jennifer Bailey have described that DCLK can mark a subset of cancer stem cells, But here we're asking the question is, can it actually give rise to pancreatic cancer as a cell of origin? And so what we've been able to show, not shown here, is that these cells are markedly expanded after cerulean. They're expanded during pan inflammation. And so we recombine then in lock-stop-locks KRAS into these DCLK1 cells using our inducible Cre-ERT mouse. And in fact, that normally they did develop pannins, so a very, very rare cell type. But most of the mice over a year developed significant pannins, but not pancreatic cancer. And of course, we then added cerulean. In fact, most of these mice then went on to develop really a very abundant pancreatic ductal adenocarcinoma. In addition, they could serve as a cell of origin for carcinogen-induced tumors, which is a difficult model. So this is DCLKs can be activated by a damaging event, and they can serve as a potent origin of cancer cells. So what I've told you so far about these quiescent pancreatic progenitors, these DCLK1 cells, is that they, under homeostasis, they really play no role. But in response to injury, they expand rapidly and lineage trace, and they also can give rise to pancreatic ductal adenocarcinoma in the setting of a KRAS mutation. So is that the only potential source of pancreatic cancer? And what, of course, I'm going to tell you is it's not, and that there are other cell types, other progenitor cells within the pancreas. And I'm just going to mention this project, which is still ongoing. This is an unpublished study, which is TFF2 as a marker of a progenitor within the pancreas. And this is a peptide. It's shown here. It's got this cloverleaf shape, the trefoil shape, and the TFF2 actually has two trefoils. And this is a family of peptides that are cloverleaf. They're small, 6 to 13 kilodaltons. And they're mainly made by mucus-secreting cells within the GI tract. So most of the publications have been on TFF2 cells in the stomach and the duodenum as a marker, for example, of metaplasia. But it's also expressed in the pancreas. In fact, the first spasmodic polypeptide was isolated from the pig pancreas. But its pancreatic expression has not been well defined. But again, what I'm going to tell you is that it marks active pancreatic progenitors. Another group, Cerathera, has actually shown it also marks pancreatic ductal glands in the setting of pancreatic cancer. So we generated this TFF2 Cre-ERT construct. We've generated one before. We made a new one to try to get better expression. And in fact, what we found was it actually had worse expression in the stomach, which was our initial target of interest. But what we found was it was very well expressed in the pancreas in a subset of single acinar cells. Not at all in the ducts. 90% were really classic acinar. There's a small subset that maybe were centroacinar. And when we isolated these cells using RFP as our marker, crossing these mice to TD tomato red, what we found was very high levels of TFF2 expression in those single isolated cells, and confirmed this by in situ hybridization, that it was acinar in location. Overlapped pretty well with MIST1 lineage using antibody staining. About 90% were co-localized with MIST1, and a very small percentage co-localized with SOX9, which is also a progenitor marker. No expression in islet cells, no expression in endothelial or mesenchymal cells. So clearly a very good acinar marker, but a unique subset of cells that express this marker TFF2. So then we asked the question, what happens to them over time? And we did lineage tracing, and in fact what we found is gradual expansion. They divided on the average every 2 months. So about 2% of the cells were labeled. Over time it rose up to perhaps 6 or 8% or more. You could begin to see, instead of single cells, occasional doublets within the pancreas. And actually, if you then did sort of confetti lineage tracing, looking for single color colonal expansion, you began to see this over the period of 6 to 12 months. And after the peak, though, at 6 months, they would begin to decline. So even though they expressed a fair amount of BRDU, compared to, for example, quiescent DCLK cells, they were not stem cells in the sense that they didn't sustain this expansion. They declined over time. So we began to think of this as a more active dividing progenitor cell, rather than a true stem cell. And I think that's a good description of this particular subset of cells within the acinar compartment. So if it's a progenitor cell that divides a little more actively than perhaps the DCLK cells, what role do they play in regeneration response to injury? And so we again used a number of different models of injury. As shown here, this is primarily cerulean, and I think in the bottom, bile duct ligation. And in the middle are elastase IL-1 beta mice. So what you see is in response to inflammation, pancreatitis, these cells really decline pretty rapidly. They really disappear, but then reemerge a month later, presumably from some other progenitor. So they don't respond very well to injury, and that distinguishes them from the more quiescent facultative progenitors within the pancreas. But if they sort of disappear in response to injury, what about, could they still be a source of cancer? And so this has been one of the debates in the cancer field, the stem cell cancer field, is, does cancer arise from these quiescent cells that don't divide a whole lot, or are they more likely to arise from progenitor cells that divide and therefore could undergo mutations? And that's still an ongoing debate. So we again examine the role of these cells with KRAS mutations. So we cross these mice, these TFF2 CREAR-T mice, to KRAS G12D mice. And what we found is, again, they developed pannins. Probably not as susceptible to pan inflammation as DCLK cells. Not shown here. It's about at 3 months, about 15%, and then 6 months, about 40% of the mice develop pannins. By 1 year, probably the majority. Not very many pannins, even though these cells are more abundant than DCLK cells. They were not quite as good at forming pannins. But on the other hand, when they did not progress to cancer, again, like the DCLK mice, but in response to cerulean-induced pancreatitis, about 30-35% of the mice went on to develop very quickly within 6 months. Pancreatitis, 100% of the mice went on within 6 months to develop pannins. So these cells, even though they are susceptible to injury, in response to cerulean, seem to be able to transition very nicely to pannins and pancreatic cancer. So what was KRAS then doing in this particular active progenitor compartment? And so what is shown here is lineage tracing with or without KRAS in response to cerulean. And so what you see is that with cerulean in wild-type mice, these cells really disappear pretty quickly, and is quantitated here. But if you put in a KRAS mutation into this sort of active progenitor, what you see is that these cells survive very well in response to cerulean. So it makes them resistant to cerulean-induced apoptosis, and they begin to sort of lineage trace very quickly, and so expand very quickly. And so I think KRAS clearly in the progenitor compartment within the pancreas has this ability to make the cells then resistant to cell death, the normal cell death that occurs with injury and transition to cancer. So here in terms of the cellular origins, I'm just going to summarize the heterogeneity of this compartment. There's both active and quiescent progenitors. The DCLK quiescent progenitors contribute very little to homeostasis, but the active TFF2 progenitors do contribute quite a bit. One is important for regeneration, the other is depleted after severe injury, but they both potentially can contribute to cancer if they acquire the mutation. So in the last few minutes, I'm going to summarize the role of nerves in cancer, something we've been working on quite a while now as well. And we all sort of know about perineural invasion and the fact that nerves are increased in the setting of cancer. Most of the focus really has been on cancer cells perhaps spreading through nerves. But it turns out that nerves also contribute signals that modulate tumor growth. And so we've been very interested in whether or not nerves could be a potential therapeutic target for pancreatic cancer. And nerves can have direct or indirect effects, and the nerves include a variety of types, including sympathetic, parasympathetic, sensory nerves. With direct effects, we're talking about receptors present on cancer cells for neurotransmitters, indirect effects acting through endothelial cells, immune cells, neuroendocrine cells, and other cell types. And I would mention that this is becoming an active area of research in general in cancer, and I'll point to nice papers recently by my colleague at Einstein, Paul Fournette, who's worked on this quite a bit, primarily in the area of prostate cancer. His lab has shown roles for both sympathetic nerves and parasympathetic nerves in prostate cancer. One may be more involved in initiation, the other more involved in metastasis, and he's focused on the role of these nerves modulating the stromal compartment, particularly, as shown in his recent paper, the endothelial compartment causing a metabolic switch within the endothelial cells, which is quite interesting. We began looking at the role of sympathetic nerves in pancreatic cancer, and with the idea that catecholamines, either circulating or released by sympathetic nerves, may play a role in progression of pancreatic cancer. And our initial observation really was this stress model, and this is, again, work done by Bernard Renz, who presented a separate study the other day. But this restraint stress model basically involves putting an animal in a mobilized stress position, actually within a sort of 50 mil Eppendorf tube with a hole cut for breathing. And the mice are very unhappy during this sort of restraint stress. But, and their catecholamine levels go up, and so this is the level of epinephrine in the blood. But it works very nice in terms of driving catecholamine signaling, and the mice also show upregulation of a specific adrenergic receptor. So what we found, really, and this is sort of the key slide, which is that in response to this sort of stress and elevation of circulating catecholamines, these KC mice, which just the KRAS mutation, normally just mostly early pannins at this early age time, progress in about 30% of the cases to full-blown pancreatic cancer, an expansion of the pannins, a huge expansion of the pannins. And this could be blocked by beta blockers. This is an ICI, it's a beta blocker drug, or adrenalectomy, removing the adrenal glands, which is the major source of circulating epinephrine, or knocking out the ADRB2 receptor. And you could actually induce cancer in KC mice by giving them infusions of isoproterenol. So this sort of slide basically illustrates that, the idea, does stress cause cancer? Well, not in a wild-type mouse, but in a mouse with a KC mutation throughout its pancreas, yes, it can. So we went on to show that using a variety of sort of drugs, here mostly, again, this drug ICI 118 551, which is a beta-2 specific adrenergic blocker, that you could prolong the survival of mice. This is actually KPC mice. So you could double their survival. You could do this with, Bernard showed, with ganglionectomy, cutting the sympathetic nerves to the pancreas. And in fact, one of the interesting things, though, was that when you blocked adrenergic signaling, not only did you see slower tumor growth and more prolonged survival, but you also saw a reduction in nerve density, as shown here. This is a staining of nerves. So why would blocking catecholamines lead to decreased nerve density? And this suggested to us that there was this feedback loop going on. And we examined this in vitro by co-culturing dorsal root ganglia with pancreatic cancer spheroids and showing this sort of crosstalk in terms of signaling, and the nerves would grow through this matrigel bridge towards the cancer cells. And in fact, this led us to recognize that the adrenergic stimulation was increasing neurotrophins, NGF, and in human cells, BDNF. And these neurotrophins, nerve growth factor, was the first growth factor described by Rita Levi Montalcini back in the 1950s. And she was looking for axonal guidance molecules, as we call them now, that caused nerves to grow out to limbs. But in fact, she isolated this from sarcoma cells and showed that the sarcoma cells were secreting a substance that caused nerves to grow into it. And actually, this growth factor is very important for both nerve growth and axonal guidance. And so overexpressing NGF, this Rita Levi Montalcini's growth factor, would actually accelerate cancer in the KPC mouse and lead to increased nerve growth. And blocking the NGF pathway using what's called a TRAC inhibitor to track the receptor for the NGF family actually led to prolonged survival and could be combined with several of the other drugs, and also led to decreased in neural density within the pancreas. So a potentially useful therapeutic drug. And Bernard went on to show in a cohort of patients in Germany that were undergoing surgery, looking at their drug history. And he first just compared whether or not they were on beta blockers, and there wasn't much of a difference. But it turns out, most beta blockers our patients are on beta-1 selective blockers. For the cardiac effects, of course, they're not interested in the beta-2 effects. And so if you just look at broad-spectrum beta blockers like propranolol and compare them to beta-1 or no beta blockers, in fact, the survival was much greater. So beta blockers do have positive effects in pancreatic cancer, but in probably many other tumors as well. So again, this summarizes the idea that stress can promote pancreatic cancer through this beta-2 adrenergic pathway. The circulating catecholamines can upregulate neurotrophins within the pancreas, which then cause the ingrowth of more nerves, particularly sympathetic nerves, which cause more catecholamine delivery. and leads to this sort of vicious feed-forward loop, this crosstalk that upregulates many growth factor pathways and can lead to progression of pancreatic cancer. And both beta blockers and TRK inhibitors prolong the survival. And as Bernard talked about yesterday, a different story of cholinergic agonists suppressing pancreatic cancer through the M1 receptor. So again, with the nervous system, there's this sort of yin and yang, promotion and suppression, tight control, really through the nervous system, which is the primary regulator of physiology in many organ systems throughout the body. So in terms of nervous and cancer, nervous signaling plays critical roles in development. This is well known, regeneration and cancer. It's important in the normal stem cell niche, something we're working on outside the pancreas as well as in the tumor microenvironment. There are, in fact, direct connections between neural terminals and stem cells, but it also, as we're finding, and as Paul Fernet has pointed out, immune responses, endothelial cells, other stromal cell types. Each tumor seems to be a little bit different in terms of its regulation. So what I've showed you about the pancreas is very different from the stomach, but we believe that this is a viable target in cancer therapy, and as I've talked about, beta-2 adrenergic receptors antagonists, M1 receptor agonists, and TRK receptor antagonists. So I'd like to stop and thank members of my laboratory and my many collaborators in the pancreatic cancer program at Columbia, and recent lab members who've really contributed wonderful science to our laboratory. Thank you very much.
It's open for questions. I'm sure there may be a few for Dr. Wang.
Yes, thank you.
Yeah, Takaori from Kyoto. Thank you very much for the very informative lecture. I learned a lot. And you have shown that the expression of SOX9 in the progenitor cell is quite limited. And as you know, several studies, including ours, have shown SOX9 positive cells are the stem cell zone of the exocrine pancreas. But very interestingly, the SOX9 expression disappears after the cell transdifferentiates into the acinar cells. So my question is about the DCLK1. I understand it's a very good marker for the progenitors of the exocrine pancreas. How is the expression pattern of DCLK1? Does it express only in the central acinar cells or in the acinar cells also?
Yeah, no, it's a good question. So the question is about DCLK1, and he commented on the overlap with SOX9, which has been shown to be a progenitor marker, and the location of these cells. And so I guess the first question is about expression, and I went over that fairly quickly. It's complicated in part because these DCLK1 cells that I described, they clearly express RNA. It's not clear they express very much protein, and as pointed out yesterday, there's multiple splice isoforms. So I don't want to be too rigid in terms of localizing this. So I would say that the DCLK cells are expressed in both the acinar, centroacinar compartment, and of course, rarely in the ducts in normal situation, but upregulated mostly in the ducts during Pannons. So many different compartments. And so how can you localize exactly which cells they are? And these DCLK cell one cells are not homogeneous themselves. There have been several subclasses. One's more inflammatory, one's more neuronal. SOX9 is a good marker, but I don't know about the heterogeneity of SOX9. So I would say, we're relying on markers to define cell identity, and I'm not sure that these markers themselves are 100% accurate. So we may be talking about all the same cells. So where is it located? I think primarily it's acinar rather than centroacinar in terms of where the regeneration is arising from. But it's very difficult to get at that. But thank you for your question, because that's really an important question in our field.
Thank you.
Thank you.
Animaj Dhar from University of Kansas Medical Center. So my question is also DCLK1, you showed the regeneration, and regeneration in PDAC can be hacked by KRAS with CMYC. Together, they can hack the regeneration. So what is the role of DCLK1 overcome that hacked program? You have any idea?
So the question is, exactly functionally, does DCLK1 play a strong role actually in this sort of regeneration process? Most of my data, I've described it simply as a marker, and I said didn't get much into the function. But the question is correct that it probably has a pretty critical function. And so studies have been done by our group, I think, with conditional DCLK1 knockout in the pancreas, which reduces pancreatic cancer. It's been done in the intestine as well. So it does have a function. And what we described, I guess, in our first paper was that DCLK1 was actually a downstream target of the KRAS program. So blocking DCLK1 blocks part of the KRAS program. So I do think it is an important functional target. We haven't looked at c-MYC, but clearly it's important for KRAS function. Other questions? Last question?
Very good talk. Thank you. Our question is, in the pioneer lesions, there are DCLK positive cells and also a lot of DCLK negative. So how can you tell differently, like which one contribute to the cancer more or less? So
again, the question is about the heterogeneity of progenitors within cancer and also in pannins, and the fact that, and this is something I think that Bob Weinberg also predicted, which is that just targeting one of these cancer stem cell populations is unlikely to be successful because of the ability of interconversion or plasticity, so to speak. Yes, so there are DCLK negative cells that can also sustain tumors, and is it because they interconvert or are they separate stem cells? I can't answer that question, but I agree that that's a confusing aspect of the field, that in fact people have described different markers, different subsets of cancer stem cells, and the exact relationship between them remains a little obscure. But thank you very much for your question. Thank you.
So Dr. Wang, thank you so much for honoring us with that wonderful lecture. I really enjoyed that. So the APA, in appreciation of your lecture for delivering the Frank Brooks State-of-the-Art lecture at the 49th Annual Meeting of the American Pancreatic Association. Thank you so much.
Thank you.
All right.
Wonderful. My pleasure.